Thank you very much for the introduction, and thank you, Professor Carraro, for the opportunity of being here to share some of our findings today. Physical exercise is well known, is well recognized of being one of the crucial modifiable factors, not only for cardiovascular and metabolic health, but also for the brain health. A primary molecular mediating these neurological benefits is the brain-derived neurotrophic factor, also known as BDNF, a neurotrophin essential for neuroplasticity, which also is involved in processes regulating cognitive function, learning, and memory. The activity of the BDNF extends beyond the central nervous system. In fact, it is highly presented in platelets, and it is also produced by the skeletal muscle tissue. In this tissue, the BDNF acts locally to support some of the muscle functions. Therefore, BDNF is a vital molecular bridge linking neurological and metabolic benefits. So much so that, in fact, circulating BDNF levels have been observed to be associated with patients with neurocognitive disorders and non-communicable chronic diseases. Building up the contraction-induced mechanism, neuromuscular electrical stimulation has been used to forcefully recruit motor units. While its role in the muscle tissue, its effect on the muscle tissue is well documented in the literature, its potential role on BDNF response remains somehow overlooked. Because there are few studies that have investigated these three variables together: exercise, neuromuscular electrical stimulation, and BDNF response. And there are fewer that have adopted functional electrical stimulation in synchrony with the voluntary activation of the muscles during the exercise. So the aim of this study was to investigate the effects of exercising with and without functional electrical stimulation in acute and chronic condition on BDNF release. To do so, we adopted a technology called adaptive functional electrical stimulation, which allowed us to trigger the electrical stimulation in synchrony with the voluntary activation of the agonist muscles of the lower limbs. And the electrical stimulation was delivered alternatively between the knee muscle extensors and flexors during the pedaling phases, during the whole training session. So moving on to the study design, we recruited 18 healthy, active men. All the participants underwent physiological evaluation to assess their maximum aerobic capacity and their peak power output. Then we randomized them into groups, a control group that only exercised, and an experimental group that exercised with the superimposed functional electrical stimulation. They both performed the same exercise protocol, which consisted in 4 bouts of 5. Can use this one. The protocol was of 5 minutes of cycling at the 60% of their individual peak power, interspersed with 3 minutes active cycling at the 40% of their individual peak power output. They followed this protocol for a total of 14 training sessions, and we also collected blood samples at the first and at the last training session at 4 time points: baseline, after 15 minutes, 24 hours, and 48 hours post-exercise. Then we also analyzed the blood. We analyzed the blood samples with specific ELISA kits to quantify the plasma BDNF. Turning to the results, no differences emerged between the 2 groups for anthropometric and principal, the main physiological variables. And regarding the acute intervention, at the first training session, the exercise elicited a marginal elevation on BDNF compared to the baseline level here, yet not statistically significant. On the other hand, the chronic intervention induced a significant BDNF response at 15 minutes post-exercise in both groups, with the control group remaining higher up to 48 hours post-exercise. Then we also compared the two conditions for each group, and the within group analysis showed that for the control group, the chronic intervention elicited a significantly higher BDNF response for all the time points post-exercise, while for the experimental group, there was a significantly high baseline level and a BDNF response after 15 minutes post-exercise. Finally, we also compared the two groups for the two conditions, and the between group analysis did not reach any statistical significance. Noteworthy is that the experimental group showed a consistent trend towards higher level of BDNF response at the 15 minutes post-exercise in both conditions and at the baseline at the last training session. So to sum up, these conclusions, to be honest, should be taken with caution due to the small sample size and the high inter-individual variability of the BDNF. However, we could conclude somehow that acute exercise elicited a marginal elevation in BDNF, yet not significant. Chronic exercise, on the other hand, elicited a significant elevation, especially in its transient 15 minutes post-exercise, and that functional electrical stimulation might act as a booster for amplifying the BDNF response comparing to the cycling alone. And one last thought is that beyond the performance, although we recruited healthy, active men, sometimes moderate to vigorous exercise intensity might be not feasible for vulnerable populations who happens to be those who might need the most for the benefits from the exercise. So maybe combining feasible intensities with the functional electrical stimulation may somehow act as a physiological amplifier to allow them to harness from the BDNF benefits and prevent any kind of risks from the higher intensities. Thank you very much, all of you, for the attention. I'd like to thank all the participants and the researchers involved. Thank you.